Molecular Genetics. UGC NET Anthropology. Part 1. Welcome to AnthroNet Academy. A study aligned with the UGC NET Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books list in anthropology. Complete with author names and insights important for UGC NET exam. Test yourself. Don't miss the final segment of this video. It includes high-yield MCQs that could make or break your next anthropology exam. 1. DNA deoxyribonucleic acid. Definition. DNA is a double-stranded helical molecule that contains the genetic instructions used in the development and functioning of all living organisms. Structure. Discovered by Watson and Crick, 1953. Composed of nucleotides, each made of a deoxyribose sugar, a phosphate group, one of 4 nitrogenous bases, purines, adenine, A, and guanine, G. Pyrimidines, thymine, T, and cytosine, C. Base pairing rule, T2H bonds, G, C, 3H bonds. Strands run antiparallel, 5 foot to 3 and 3 to 5. Functions. Storage of genetic information. Replication for cell division. Template for RNA synthesis, transcription. 2. RNA, ribonucleic acid. Definition. RNA is a single-stranded molecule that helps in the expression of genetic information stored in DNA. Types of RNA. mRNA, messenger RNA carries genetic code from DNA to ribosomes. tRNA, transfer RNA brings amino acids during protein synthesis. rRNA, ribosomal RNA, structural and catalytic part of ribosomes. snRNA, miRNA, siRNA, involved in gene regulation and splicing. Structure. Sugar, ribose. Bases, AU, uracil, GC note. Uracil replaces thymine. Usually single-stranded, but can form secondary structures, hairpins. Functions. Transcription, mRNA synthesis from DNA. Translation, decoding mRNA to synthesize proteins. Regulation, gene expression and RNA interference, RNAi. 3. Genetic code. Definition. The genetic code is the set of rules by which the sequence of nucleotides in DNA or RNA is translated into the sequence of amino acids in proteins. Key features. Triplet code. Each amino acid is coded by a sequence of 3 nucleotides, codon. Degenerate. Multiple codons may code for the same amino acid, e.g., LU, UUA, UUG, CU, CUC, CUA, CUG. Unambiguous. One codon specifies only one amino acid. Universal. Same in nearly all organisms. Start codon. August codes for methionine, signals start of translation. Stop codons, UAA, UG, UGA, do not code for any amino acid. 4. Protein structure and synthesis. A. Protein structure. Primary structure. Sequence of amino acids in a polypeptide. Held by peptide bonds. Secondary structure. Local folding, alpha helix, beta sheet. Stabilized by hydrogen bonds. Tertiary structure. 3D folding of a polypeptide. Stabilized by ionic, hydrogen, disulfide bonds and hydrophobic interactions. Quaternary structure. Association of multiple polypeptide chains. Example, hemoglobin, 4 subunits. B. Protein synthesis, central dogma. Transcription, DNA to mRNA. Definition. Transcription is the process of synthesizing a complementary RNA strand from a DNA template. This RNA is typically mRNA. Messenger RNA, which carries the genetic code to the ribosomes. Location. Eukaryotes, nucleus. Prokaryotes, cytoplasm, no nucleus. 1. Initiation. RNA polymerase binds to the promoter region, a specific sequence upstream of the gene, e.g., TATA box in eukaryotes. In prokaryotes, a sigma factor helps RNA polymerase recognize the promoter. In eukaryotes, transcription factors and RNA polymerase II form a transcription initiation complex. DNA unwinds at the promoter region, exposing the template strand, antisense strand, for transcription. 2. Elongation. RNA polymerase moves along the template strand in the 3'5" direction. Synthesizes RNA in the 5'3" direction by adding complementary RNA nucleotides. A U, TA, GC, CG. As the enzyme moves forward, the DNA helix rewinds behind it, and the growing RNA strand peels off. 3. Termination. In prokaryotes, row-independent termination forms a hairpin loop in RNA that destabilizes the complex. Row-dependent, a row protein helps detach the RNA transcript. In eukaryotes, termination occurs after the polyadenylation signal, OAO, is transcribed. RNA polymerase continues a bit further, then the transcript is cleaved and released. 4. Post-transcriptional modifications, eukaryotes only. 5' capping, addition of 7-methylguanosine to the 5' and protects mRNA and helps with ribosome binding. 3' polyadenylation, addition of a poly-A tail, 200 adenines, stabilizes mRNA. Splicing. Introns, non-coding regions, are removed. Exons, coding regions, are joined. Performed by spliceosome complex. 2. Translation, mRNA to protein. Definition. Translation is the process by which ribosomes synthesize a polypeptide protein using the codons in mRNA as a template. Location. Cytoplasm in both prokaryotes and eukaryotes. In eukaryotes, ribosomes may be free-floating or bound to rough ER. Steps of translation. 1. Activation of amino acids pre-step. Each amino acid is attached to its specific tRNA by an enzyme called aminoacyl tRNA synthetase. This enzyme ensures charging of tRNA with the correct amino acid requires ADP. The tRNA has an anticodon that pairs with the codon in mRNA. 2. Initiation. The small ribosomal subunit, 40S in eukaryotes, 30S in prokaryotes, binds to the 5' end of mRNA. It scans for the start codon August, which codes for methionine. The initiator tRNA, metrona, binds to August. The large ribosomal subunit, 60S in eukaryotes, 50S in prokaryotes, joins to form the complete ribosome, 80S in eukaryotes, 70S in prokaryotes. The ribosome has 3 binding sites. A site, aminoacyl accepts incoming tRNA. P site, peptidyl holds growing peptide. E site, exit releases empty tRNA. 3. Elongation. New aminoacyl tRNA enters the A site. A peptide bond forms between the amino acid in the P site and the one in the A site, catalyzed by peptidyl transferase, an rRNA-based ribozyme. The ribosome moves, translocates, 1 codon forward. tRNA in P site E site exits. tRNA in a site P site. New tRNA comes into a site. This cycle repeats, and the polypeptide chain grows and terminus to C-terminus. 4. Termination. When the ribosome reaches a stop codon, UAA, UG, or UGA, no tRNA binds. A release factor binds instead, causing the ribosome to release the polypeptide. The ribosome dissociates into subunits and releases the mRNA. 5. Post-translation modifications. After synthesis, the protein may undergo folding, often aided by chaperone proteins. Cleavage, e.g., insulin. Phosphorylation, glycosylation, methylation, etc. Transport to correct cellular location. Summary chart, transcription versus translation. Feature transcription translation. Template DNA mRNA. Product mRNA protein, polypeptide. Location nucleus, eukaryotes, cytoplasm, proc. Cytoplasm, both. Enzyme RNA polymerase ribosome, rRNA plus protein complex. Start site promoter start codon, August. Termination signal terminator sequence stop codon, UAA, UG, UGA. Post-processing capping, tailing, splicing, eukaryotes, folding modifications. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.